Hello, everyone. Welcome to today's live webcast, Way More Biologic Stability, A Lot Less Protein. Today's webcast is being sponsored by Unchained Labs, who is committed to building the first cool biologics tools company, one with products that will make a real difference in the research scientists do every day, and one that helps biologics researchers break free from suboptimal tools. And now for some important announcements. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them into the Q&A box, which can be found by clicking on the red Q&A widget at the bottom of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during this event. If you have any technical problems with viewing or hearing this presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. And now I would like to introduce today's speaker. Presenting today is Dr. Dan Lund, a scientist at Unchained Labs. Dan has more than 12 years of experience biophysically characterizing diverse, novel protein molecules. For the last 7 years, he has been involved in the development and marketing of the most versatile stability characterization and screening platform, the UNIT, which lets researchers look at more protein stability parameters than any other tool out there. He is currently the field application scientist in Europe for Unchained Labs, covering a range of cool and innovative protein stability technologies. And thank you, everyone, for joining us today. Dan, why don't you go ahead and get us started?
Thank you, Sarah, and thank you to everyone else for joining our presentation today. I hope you're having a good day, whether it's morning, afternoon, or evening, wherever you are in the world. And welcome to the webinar from Unchained Labs today, which is about biological stability and ways you can get a lot more information using a lot less protein. So for those of you who aren't familiar with us at Unchained Labs, what are we all about? Well, I think Sarah mentioned this already, but we're all about breaking free. So everybody wants to break free from something, right? So I'm in the UK at the moment, and the weather is absolutely awful, so I'd love to break free from all the rain and all the horrendous darkness there is over here. That's just me. In Unchained Labs, we're more about helping biologic researchers break free from suboptimal tools. So a lot of the tools that we're using in the lab these days to characterize our biological molecules are designed for something other than biologics. So they may be designed for chemicals, for paints, or for small molecules. We don't care about any of that. We're only about biologics. So we have a range of useful scientific tools which enable you to understand your biologic better, whether it be in terms of screening, predicting aggregation, or determining size characteristics. So what are biologics? So the antibody has been around for a number of years now. The first one was approved by the FDA in around 1986. And since the year 2000, the therapeutic market's grown exponentially. There are at least 30 FDA-approved therapeutic mAbs available now. Yet mAbs are not perfect. We're seeing a lot of novel formats of biologics going through various stages of development, whether they be Fabs, fusion proteins such as single-chain variable fragments, or heavy-chain antibodies. And of course, there's antibody-drug conjugates, which combine the specific targeting capabilities of antibodies with a highly cytotoxic drug. So we're all faced with challenges of developing these novel proteins. We're also faced with challenges improving the patient experience. So we're seeing two trends in biologic development these days. Seeing a trend towards higher concentrations of biologic drugs so that patients can self-administer more easily at home. And we're also seeing a trend towards screening of higher candidate numbers. So we want to characterize better different mutations or variants of our proteins in order to have a better idea of what we're seeing. But all this requires more screening, and it all requires it with less samples. So what's the problem? Why is this so complicated? Well, we want to maximize the efficiency of our biologic drugs, which means we have to do two things. We need to minimize the amount of inactive, unfolded protein that's there at delivery, and also minimize aggregates, which may also be inactive, but more importantly, it might lead to an immune response. The complication arises because proteins are only marginally stable. They have an intrinsic stability which is not very high. So there's lots of different ways that proteins can degrade. Unfortunately, in order to manufacture, to transport and to deliver protein therapeutic drugs, there are a lot of stresses that proteins are subjected to. So the way that we need to get around these problems and these different stresses is by optimizing various stages. We can optimize the process of production, we can optimize the protein itself, and engineer different constructs in order to intrinsically improve the stability, or we can optimize the formulation. The first step in this process is to determine our degradation pattern. So there are multiple ways of proteins degrading. It could be unfolding, it could be aggregation, fragmentation, or chemical modification. And all of these are going to lead to characteristics which are unfavorable. Now, sometimes we don't know what the degradation pattern is. So when we're developing monoclonal antibodies, especially if we've already done it in the past and we're just making small changes to the CDR, for example, then we often can't have an idea of what's going on with novel proteins. With MABs, we can do it more easily. We can use a platform approach potentially, and that's often good enough. But with the new platforms, any methods of predicting the optimal conditions that we have has significant value. I think we might be having a technical difficulty, so if I can just ask you to bear with us for a second. Okay. So, how might we go about predicting the optimal conditions of stability? There are multiple ways that this has been done in the past. Melting temperature is one which is familiar to a lot of you, and we've all done these kind of experiments. Aggregation onset temperatures can help us to characterize aggregation propensities. We can optimize against viscosity and injectabilities of proteins, if we can determine that viscosity. And also, if we can look at various other measures of protein-protein interactions, whether that be through a B22 parameter, which is a second virial coefficient, fusion interaction parameter, or various others. And these are just the physical or the solution properties that we need to consider. But it's not always so simple. The landscape is more complicated, and there are also chemical conditions that we need to determine also, such as fragmentation, oxidation, glycosylation patterns in the different glycoforms, and how they may affect the stability of our proteins. Today we're just going to focus on two. We're going to focus on the solution properties and the physical stability, because they impact quite significantly in a large deal of our systems. So fundamentally, what do we need to do? In order to have an efficient and effective biologic drug, we need to maximize the population and the amount of the functional form, so the native protein. We don't want any denatured protein, because that won't have the correct function. It won't have the correct form in order to perform its task. And we don't want any aggregated protein, whether they be native state aggregates or whether they be denatured state aggregates. So we need to do some clever planning and some clever engineering in order to make sure that we maximize the concentration of that native form of protein. And this isn't always easy. It is challenging, and sometimes, especially if we don't know what's happening, designing our processes or formulations is challenging and can require a leap of faith. So we may stress the protein via a particular mechanism. We may wait for weeks, wait for months even, and characterize what happens. But if we could have some way of predicting the stability early on, that would be a very advantageous way of being able to develop our biologics. What we would want to do is be able to predict what the stability was during those various stages of either manufacturing and transport, during long-term storage, so we're talking drug product and drug substance here. And the benefits of doing this early in development is we reduce our development risk, we accelerate our development process, potentially increase our manufacturing yield and lower costs, and generally lead to a better product. Essentially then what we have is that if we do more candidates and investigate more formulations, we get more information about each one, then that gives us the best chance of identifying early in development the optimum candidates and or formulation. And equally as important, of identifying those which are likely to fail so that we can stop wasting time and not develop them. But the benefits of early stage stability screening are clear. But there are challenges involved as well. So there are lots of samples that we need to investigate. We never really have enough protein to do all the measurements and investigations that we think we should. And also, it's difficult sometimes to have investigations which help us understand what's happening and that are predictive. And that's what we need. Well, there is a tool that can help you do that. It's called the Unit. It's available from Unchained Labs, and it's the most powerful biologic stability screener. It allows you to measure stability through any form of thermal denaturation, simultaneously determining aggregation and conformational characteristics, such as the thermal melting temperature or the aggregation onset temperature. It requires only 9 microliters of sample. The samples are sealed, so there is no evaporation. We obtain a full spectrum of fluorescence and static light scattering. It's an optical technique. It allows label-free detection, yet you can expand the applications available by adding dyes if you need to probe different aspects of stability and different indicators. The system is high throughput. It allows 48 samples per run to be measured. The samples are stored in quartz cuvettes, measured simultaneously and heated at the same time. The heating is very uniform and there is no cross-contamination. The samples are stored within a proprietary cuvette system called the Uni. The Uni allows us to use 9 microliter sample sizes. There is no limit to the type of sample that you can measure. We can measure intrinsic fluorescence and the fluorescence of extrinsic probes and measure light scattering simultaneously. There is even sample heating, and the Uni is what makes the Unit truly unique, providing the throughput and the small sample size. Sample preparation and loading is extremely straightforward. Samples can be prepared in a 384 well plate and transferred across to the Uni, either using a multi-channel pipette or by using individual pipettes. The samples are then sealed to prevent evaporation or any sort of concentration changes related to that during the sample heating. So I mentioned that we are using an optical technique to follow the conformational changes and the aggregation effects of proteins as they are heated. So this is a typical trace for a monoclonal antibody. You will see on the left-hand side we have the blue text, which represents the unfolding of the protein. As the number goes from low values to high values, what we are seeing is a shift in the wavelength of tryptophan fluorescence as the polar environment goes from a buried to a polar exposed state. And from that we can characterize melting temperatures by looking at the midpoints of the steps in the transitions. Simultaneously, by acquiring static light scattering data, we can characterize the aggregation. So in this case, you can see aggregation does not occur until the second melting event. So we can correlate aggregation of this protein and thermal denaturation as being related to that particular domain. From the literature on this protein, we know that that domain corresponds to the Fab region, that part of the protein that drives the degradation. What's the raw data of the system like, and how is this reflecting, and what are the advantages of this mechanism? Well, we obtain a spectrum. You can see an example of raw data here. There are 3 peaks on the signal. The first represents the static light scattering at 266 nanometers. We then have fluorescence from the aromatic amino acids within the protein. Typically, tryptophan and tyrosine dominate this signal. There is then a second static light scattering signal at 473 nanometers. By looking at the whole spectrum, it allows us to account for different variations of protein unfolding patterns. And by monitoring aggregation at 2 wavelengths, we can follow different aggregation mechanisms. So how might changes in this signal reflect changes in conformation of the protein? Well, it depends on the protein, and they all behave differently. So on this slide, you can see 4 figures. MAB1 in the top left, we see similar intensities but a shift from lower to higher wavelengths as the protein unfolds. MAB2 also has a shift, but we also see an intensity drop. MAB3, there is no shift, but the intensity decreases significantly and disappears. And with the 4th protein, cytochrome C, we see initially no fluorescence when the protein is folded due to a heme group which quenches the fluorescence. But then on unfolding, the heme group moves away from the tryptophan which is there, and we see a fluorescent signal. So different ways that proteins can be characterized, all done by measuring a full spectrum. We look at the example of cytochrome C. See that if we try and follow a shift in the peak, in this case we're using a ratio between the intensities at 2 different wavelengths, 330 and 330 nanometers, we don't get a very good melting curve. But if we look at the intensity distribution, we see a very clear transition and get very high reproducibility and accuracy on our melting transition. With static light scattering in the system, this is a proven, well-characterized way of characterizing protein aggregate detection. We have two lasers to look at static light scattering. We have a 266 nanometer laser, which is primarily there to excite the intrinsic fluorescence of the protein, but also at low concentrations is useful characterizing very small aggregates, low protein concentrations, and it's very highly sensitive. At higher concentrations and with larger aggregates, at the point when that signal begins to saturate, we have a second laser system which enables us to characterize concentrations up to hundreds of mg per mil, very high sizes of aggregates, and with a large dynamic range. How is this reflected in a thermal unfolding experiment? Well, because we know that as you unfold a protein with temperature, polydispersity is high, aggregate formation is extremely random. We do have both large and small aggregates which coexist. With the 266 nanometer laser, you can see we have an aggregation onset temperature, Tag, which occurs around 64 degrees, whereas with the 473 nanometer laser, we don't see the aggregation occurring till later. So we're sensitive to small aggregate formation with the 266 nanometer laser and very sensitive, and with the larger wavelength laser, we can see larger aggregates. With a single wavelength system, you get an incomplete picture of stability. Everything I've spoken about so far has been related to thermal ramping, but that's not the only way that you can use the unit to characterize stability of your proteins. Essentially, you can do anything you like. If you can draw a thermal degradation pattern, then you can do it. So you can look at isothermal characteristics, thermal recovery, and other different mechanisms as well. In terms of the isothermal stability, what are we looking at here? Well, in this figure, we have on the y-axis, we're looking at the unfolding of this is a Fab protein. This is at 65 degrees in different concentrations of sodium chloride. And you can see over the course of 10 hours, different concentrations of salt lead to the protein unfolding at different rates. Similarly, we look at the aggregation of a protein. We see in this case, we have a monoclonal antibody with different amounts of arginine. So comparing the aggregation of IgG at 65 degrees in the presence of the increasing amounts of arginine over an 8-hour time course allows us to show that 10 millimolar arginine in this case is insufficient to suppress aggregation. That's the yellow curve. 100 millimolar arginine significantly affects aggregation, and that's the purple curve. And 500 millimolar completely suppresses the aggregation. And experiments of this duration are only possible because we seal our samples in. We seal the unis. So determining kinetics is a useful way of determining potential degradation optimization mechanism. With thermal recovery, in this case of an IgG, we're able to heat the sample up to an elevated temperature and cool it back down. We can see how much of the protein recovers its initial conformation. Because we're using a full spectrum fluorescence, we're able to see very small differences in that fluorescence spectrum, indicating small differences in the tertiary structure. An example of how this might be used. We have in this case again the unfolding and the aggregation of an IgG. This time we've done multi-cycle thermal recovery. The unfolding represented on the top panel and the aggregation on the bottom panel. In the presence of, in the blue curve, is at 500 millimolar arginine, and in the absence of any arginine, just the IgG on its own. So arginine is very well known to stabilize the denatured state of the protein. So, in the presence of arginine, as we heat the protein up, you can see that the denatured state is more stabilized, the protein unfolds more, and when we recool it, it doesn't recover back to its original state, but it does recover to a baseline level. In the absence of arginine, there's very little unfolding until we increase the temperature up to 70 degrees. That's the right-hand side of these figures. And you can see then that the unfolding continues. Now, if we look at the other side of the story, the aggregation. In the bottom panel, you can see in the presence of arginine, there is no aggregation at all. We're not seeing any aggregation occurring, even when we increase the temperature to 70 degrees. But in the absence of the arginine, we're seeing significant aggregation. And even after only 2 or 3 cycles, we're starting to get irrecoverable aggregation occurring. So this thermal recovery assay allows you to see the effects of different formulations have on both the unfolding and the aggregation. And only by studying both the melting and aggregation simultaneously can we see the entire picture of what is happening to the protein. Up to now, I've only discussed the intrinsic fluorescence and the aggregation that you can measure with the unit system. But we also offer greater flexibility with 2 additional systems by looking at the effects of different probe dyes to characterize stability in different ways. First of all, we have a unit with an optional 445 nanometer laser. This allows us to determine relative viscosities of protein formulations. The way we do this is we have a molecular rotor dye. The dye absorbs energy from a laser and it dissipates it in one of 2 ways. The dye has a bond which can rotate. If it is in a low viscosity solution, the bond rotates freely and energy is dissipated through that rotation. As viscosity increases, the rotation is slowed, so the energy has to be dissipated through another mechanism, and that's through fluorescence. So what we see with this dye, which is called CCVJ, as viscosity increases, we see a linear increase in the fluorescence signal. This allows us to characterize viscosities in the absence of shear, which is important for characterizing potentially injectables or for various processing steps, and also as a measure of protein-protein interactions. It agrees very well with a viscometer. In this case, we have data of increasing dextran concentration measured on a commercial rheometer system, where we've measured the actual viscosity, then characterized that against the viscosity of the fluorescence of our viscosity-sensitive dye, and you can see that the correlation is extremely good. So we can now, in the same experiment, measure viscosity of our formulation samples, and then characterize thermal unfolding measurements. How is this useful? Well, commonly we know that certain excipients can be used to reduce viscosity. In the left-hand panel, you can see different concentrations of potassium iodide lead to a reduction. As you increase the concentration, we see a reduction in the viscosity measurement on the unit system. On the right-hand side, changing the ionic species according to their order in the Hofmeister series, we see also a reduction in the viscosity. These are both well publicized events and publicized characteristics, and the measurements that we obtain on the unit system agree very well with what we see in the literature. Also, consider the effect that you have a protein where there is no tryptophan, or the tryptophan environment doesn't change significantly. Now, typically you may introduce a dye to measure this kind of system. So we have another system which characterizes proteins where standard dyes do not work. So consider the example of a membrane protein. It may be that the tryptophan in the protein is encased within the membrane, so when the protein unfolds, the environment does not change. It may also be that the proteins are in solution rather than in a membrane, but are stabilized by the presence of surfactants at high concentration. Traditional dyes such as cypro-orange, which interact with hydrophobic regions of proteins, will interact with the surfactants as well, and will give a high baseline signal, making it very difficult to obtain an unfolding characteristic. The assay we have in our unit with 375 nanometer laser system uses a dye called CPM, which covalently binds to thiols and increases its fluorescence as thiols are exposed. Now, these thiols may be part of cysteine residues, which are free, or it may be part of a disulfide bond which breaks as the protein unfolds. Typically what you would see in this case is as you increase the temperature, you increase the signal from the dye. This allows us to screen conditions for drug targets, potentially optimizing crystallization or also just optimizing stability. And again, it's only by using the full spectrum fluorescence that we're able to do this. So that's information about why we are doing and what the interest is in high throughput early stage screening of proteins and the unit system and what it can do. I'm now going to go over 4 case studies about examples of how the system is used and how it can be used for predicting various different kinds of things. So I'm going to start case study number 1, where we're going to be screening for long-term storage, optimizing that for a monoclonal antibody. So I wanted to describe to you an experiment that we have done at Unchained Labs in-house, where we've looked at a monoclonal antibody which we were kindly gifted by a commercial partner. And we wanted to see whether or not experiments and measurements that we obtained were predictive of long-term stability. So we took our monoclonal antibody, we put it into 144 formulations with a wide range of different variables, and we measured the stability in one day of all those 144 on the unit system. We then took a subset of those formulations, we took 12, and we put them into a 6-month stability study. We incubated them at 40 degrees, and we characterized at various time points what the degradation of the protein was through various mechanisms, both at the unit, with size exclusion chromatography, and with DLS. So the initial day, we wanted to know whether or not our prediction correlated with that long-term storage stability. 144 samples, so 1 day of work to measure and analyze all of these samples. You can see there's a vast amount of data with a wide range of different patterns and different conformational stabilities. We have on the left-hand side information relating to the conformation, giving us the melting temperatures, and on the right-hand side, information relating to the aggregation. So aggregation onset temperatures, but also amplitude changes, so the final value, how aggregated the protein becomes. When we take a subset of those 12, now the subset we took, it was not the best 12, it was not the worst 12, it was 12 formulations which had been used on commercial products at various stages. And we looked at those, and you can see just on here that there is quite a wide range of differences between the different formulations. So focusing on the left-hand side, you can see there are different melting temperatures for the different proteins, indicated by the different steps. But also you can see that the value at 15 degrees also differs between formulations. Green curve starts at a significantly higher value. The orange curve starts at a slightly higher value. So just by changing the formulation, we're changing the conformation itself of the protein. On the right-hand side, we have the aggregation data. Again, you can see some of the formulations aggregated at different temperatures. They reached final endpoint values, and you can see that the order at which these things happened in the aggregation is not the same order as it happened in the unfolding. So what did we see? Well, what we did was we measured different buffers, including glycine, citrate, acetate, and tris. We measured a range of pHs from 2.2 to 10.6, indicated by the dashed bars on the two graphs on the slide. We measured a low ionic strength sample at low pH. And also we measured different additives as stabilizing excipients, which are typically used in formulation, including trehalose, polysorbate, and arginine glutamate. On the top panel, you can see we have black squares corresponding to the first melting transition. We have red squares corresponding to the second. And you can see that there are differences between the samples and the formulations at different pH, and also within the same pH in the presence of different excipients. In the bottom panel, you can see there are black dots representing the aggregation onset temperatures. Again, you can see that there are differences between all the different formulations. In the first two formulations itself, you can see there was no aggregation observed anyway. So what did we see? Did this data that we measured correlate with what happened over the long period of time? Well, yes, it did. So we took those melting temperatures for a particular part of the protein, so for the Fab domain, that we measured in 1 day, and we correlated those with the rank order of how much protein was left measured on SEC at 24 weeks after it had been stored at 40 degrees. And you can see the correlation is outstanding. So the value here, we can measure in 2 hours data which gives us an indicator of protein stability versus 6 months with far less protein consumed. Now obviously we're going to be doing accelerated stability with SEC anyway, but the value of these measurements is that it allows us to remove those formulations which we may have had to measure anyway, but now we can see that they're going to be very poor in terms of stability, so we can save ourselves significant time. The second case study doesn't look at long-term stability. It looks at screening for developability. So rather than necessarily screening for storage, we're screening for how developable the proteins will be. And the particular case study that I'm going to show you involves subjecting the monoclonal antibodies to low pH. So making MABs requires low pH. whether it be in a protein A elution step, where you have to use pH 2.5 to 4.5 to remove the antibody from the protein A, or whether it be in a viral clearance step. And the experiment design we had, so it was a similar data set to our previous case study. It was the same monoclonal antibody. We had our samples at pH 3.6 in the presence of a little bit of salt and trehalose and 50 millimolar of buffer. The only thing we changed here was the buffer species. So we used both acetate, which is represented by the dark blue traces on the graphs in the figures, and we used citrate, which is represented by the light blue traces. The only difference between these two molecules is that acetate is monovalent, whereas citrate is trivalent. So what we saw was that in the presence of citrate buffer, we had a lower conformational stability. Changing to acetate increased both melting temperatures for both transitions. And also we had a higher aggregation propensity in citrate. You can see the dark blue curve for acetate has a higher aggregation onset temperature and also reaches a lower final value. There is less aggregate forming. So our prediction then is that citrate is a worse buffer at low pH than acetate for this protein. So what we've done next is we've looked at, again, similar conditions. So we've taken these two to determine whether or not that is the case in longer term. So on all these figures, the open markers represent protein which has been stored at 4 degrees. The closed markers represent protein which has been stored at 40 degrees. Now in the two graphs on the slide, at the top panel we are looking at an isothermal experiment on the unit just at 25 degrees, and we're measuring the initial fluorescence peak position. We're not doing any thermal denaturation, we're simply looking at the conformational state of the protein at that time. You can see in the citrate, so the light blue curve, reaches a higher value. There is a larger change in that conformational state over a period of time. That indicates that in citrate, the protein isn't folding, more so than it is in acetate. The bottom graph, the bottom panel, you can see we're looking at the light scattering data. All the graphs, all the traces are pretty flat, with the exception of citrate, which you can see increases dramatically. So even after a period of 4 weeks, it's got a huge light scattering signal compared to all the others. This indicates that the protein is aggregating significantly. How does this compare with more traditional ways of characterizing these parameters? Well, again, we've looked at monomer depletion using SEC. And on the top right panel, you can see the solid markers for the dark blue trace representing acetate. There is some depletion. We are seeing some aggregation. But with the citrate, we're seeing complete loss of the monomer after only 4 weeks. Again, looking at the change in concentration, you can see that the concentration changes dramatically for citrate. So after 12 weeks, that's down to 0.2 mg per mol from a starting value of around 1, indicating that a lot of the protein has dropped from solution. The advantage for using the unit for these kinds of measurements is that there is no need to filter or centrifuge the samples. We can measure everything that's in there. Also, they only require 9 microliters, and the samples can be recovered. Moving on to the third case study. We're going to look at antibody-drug conjugates. We're going to characterize their stability and their formulation. So antibody-drug conjugates are significantly more complicated than monoclonal antibodies. For a start, they have a very large hydrophobic drug attached, so the stability is going to be affected in different ways. On this graph, we have two traces, two types of trace. Traces represented by the dots represent the conformational change of the protein, so the shift in the fluorescence peak position. Those represented by the lines represent the light scattering, so the aggregation of the protein. Now, if we initially look at the light scattering, so if you focus on the very bottom right-hand corner of this graph, you will see a red trace very close to the axis, and that is the protein on its own. That is MAB. That is trastuzumab, and that is with no drug attached, so you can see very little aggregation. However, as soon as you start attaching drugs to this antibody, you can see that the light scattering starts to increase significantly between 70 and 80 degrees. There's also an effect on the conformation. If you look at the red dot trace representing the protein alone, you can see there are 2 folding transitions. As you add drug to the protein, you can see you're destabilizing principally the first protein transition and moving that down to lower temperatures around 62 degrees. To an extent, that's to be expected. But even the choice of drug can significantly affect the stability of the ADC. It's not just that. We want to measure, or we want to make ADCs which are as highly loaded in drug as possible in order to deliver the best therapeutic effect at the lowest dose. So in this case, we have trastuzumab again with different amounts of drug attached, so a different drug-antibody ratio, or DAR. Again, focusing on the light scattering data in the bottom right corner, you can see the drug alone aggregates at around 83 degrees. Then as you start to add more drug, the antibody alone aggregates around 83. And as you start to add more drug, you can see that not only does the temperature move to lower temperatures, the aggregation onset temperature moves to lower temperatures, but the actual amount of aggregation that occurs increases as well. So that's to be kind of expected. These drugs which we're attaching to our antibodies are very hydrophobic, and the more hydrophobic patches that there are on the surface, the more likely that the protein is to aggregate. We're also seeing a destabilization of the protein as we add the drug, and you can see by looking at the dots again, the conformational changes. The red dots represent the protein on its own, and as you add more and more drug, you can see principally the first transition starts to decrease in its melting temperature. That is potentially indicating that the drug itself is binding to the region of the protein indicated by that first transition. Finally, for ADCs, formulation is critical. A lot of antibodies over the years have been developed and have been formulated as a platform in PBS, and it's been very successful. Unfortunately, the same does not apply with ADCs. If we look on this trace, you can see the light purple traces representing the drug, the ADC in PBS. You can see at around about 70 degrees, we see a huge decrease in both of those signals. That's indicating that the protein is dropping out of solution. The darker blue traces represent the protein in sodium succinimide, and you can see that the protein, while it does aggregate, it aggregates at a higher temperature, and it does not precipitate either. So sodium succinimide is definitely a better formulation for this drug than PBS. Final case study is a slightly different way of looking at things. Everything we've done up to now has either been looking at the light scattering and the conformational fluorescence signal, either by doing isothermal experiments at elevated temperatures or by doing thermal unfolding. Now we're going to look at a different mechanism for stabilizing proteins. We are going to look at stabilizing the native state. And we're going to use just the light scattering here. So I mentioned earlier on about the second virial coefficient. Well, what is that? What is that and how does it work? Well, the idea is that at low concentrations of protein, the signal that you get from light scattering is proportional to the concentration multiplied by the size. And that's great. But that only applies if you have a monodisperse, a random dispersion of molecules in solution. If you have a non-random distribution, then that starts to break down. And if you look at the left-hand panel, you can see that while we have an equilibrium at the top of that panel between native protein and unfolded protein, all randomly distributed in solution, as long as we have that arrangement, we will see a linear increase with our light scattering signal with protein concentration. If there are interactions between the proteins, let's say that there are attractive interactions between the proteins caused by weak long-range electrostatic forces, for example, then we no longer have a non-random distribution of proteins. Under that situation, the light scattering equation alters. We no longer have that the intensity is directly proportional to the product of the concentration and the size. There is also extra terms involved depending on whether it is an attractive or a repulsive interaction. If the interaction is attractive, the light scattering signal is higher than we expect it to be at higher concentrations of protein. And if the attraction is repulsive, then the light scattering signal we measure at higher concentrations is lower than we expect it to be. So in this article published by Hoffman et al., what they've done is characterized using the unit system between 0 and 10 grams per liter, they have measured the light scattering signal and determined a linear fit. They then extrapolated that up to higher concentrations. They then measured directly at 40 mg per mil in this case, and where they see higher than what they expect, then they determine an attractive interaction, and where they see lower than what they expect, they determine a repulsive interaction. And for this particular Fc fusion protein, they expect that if they have attractive interactions, then they're more likely to see aggregation. So what did they see? Well, in this case, they did a number of different formulations, and they characterized and calculated the difference from that linear fit, so whether it was different from the blue line on the previous slide, whether it was positively different, or whether it was negatively different. And they did that for a range of formulations. So for this particular Fc fusion protein, you can see at pH 7, there is a large deviation, a large positive deviation from that linearity. But simply by adding 100 millimolar salt, they're able to bring that within an acceptable region for them. At pH 5.5, you can see there's a large negative deviation from linearity, indicating repulsive interactions. And when they screened their different proteins in this mechanism, it took them very little time. It was a simple 3-minute experiment that they did. And this correlated with their long-term turbidity experiments, indicating that this was for them a good way of predicting their protein stability. So as I hope you can tell, and I hope you can gather from what we've talked about today, there are a wide variety of different assays and different mechanisms that you can use to determine protein stability with very small amounts of sample, very quickly and very easily with the unit system. So in terms of different applications that are available, you can characterize melting temperatures, aggregation temperatures, look at thermal recovery and isothermal stability, as well as look at relative viscosity and the soluble membrane protein stability using one of 3 different unit systems available from Unchained Labs. And by looking at so many different types of protein stability, it allows you to build up a big picture of what your protein is doing. Rather than focusing on an individual small aspect of that protein, you're able to build up a bigger picture. And that's why we at Unchained Labs, we provide the right tools to do the right jobs. We provide tools that allow you to characterize protein stability in terms of screening, predicting aggregation, and determining size. And with that, I'd like to thank you all for listening and invite any questions.
Thank you very much, Dan. And we do have quite a few questions in here for you. The first question that we have here, the measurements of viscosity that you have, which use a molecular rotor, they appear to be done in the absence of shear. Does that mean that your viscosity isn't useful as a measurement of injectability?
So that's right. Our measurements are done in the absence of shear. So if you have a protein that shear thins or shear thickens, then the measurements that we obtain don't give a direct measurement of the injection force required, for example, so whether it's for delivery through a needle or to achieve an appropriate flux through a TFF step, for example. But don't forget we're screening. So changes in viscosity that are caused by differences in formulations or different excipients at 0 shear are likely to have a comparable effect under shear, even if the absolute viscosity is different. Also, we're looking at viscosity as a potential indicator of protein-protein interaction as well. So 2 different ways of using viscosity measurements.
Thank you. And when do you use emission ratios versus emission intensity to evaluate TM?
It depends. So sometimes you don't necessarily know what structural characteristic is going to happen when you heat your protein. So sometimes if you have a protein where there is quenching in the native state, then you won't be able to see any signal at all when the protein is folded. So you won't be able to follow that unfolding using a ratio change. You would have to use a change in the intensity.
And how does the TM data generated with UNIT compare with that by DSC?
It's very similar. We're looking at a slightly different physical effect, obviously. We're looking at changes in polar environments around tryptophans, whereas DSC is looking at the energy flow through the hydration layer around the protein. So because we're looking at more specific local interactions, we tend to be a little bit more sensitive But the rank orders are all the same. There may be 0.2, 0.3 degree difference. The fluorescence will typically give a slightly lower TM, but pretty much identical. And certainly in terms of screening, there's no difference in which mechanism is more applicable. Just our uses much less sample. It's much faster, and we also tell you about the aggregation.
Wonderful. And what is the protein concentration for TM and tag measurements?
It's about the same. So obviously it's protein dependent, but normally we say your safe working range is about 0.05 grams per liter of protein up to that. Well, the highest we've done is 150 mg per mil. Some of our customers are using higher, but we've never managed to solubilize proteins at that concentration. So very broad dynamic range, and very, very easy to do in the same experiment as well. You don't necessarily need to do different experiments for different concentration ranges.
Excellent. And does the unit measure absolute viscosity in steady poise or just a relative viscosity?
So at the moment, we're just measuring a relative viscosity. It's possible, I imagine, to do absolute viscosity with the creation of an appropriate standard curve, and this is something that we plan to do in the future. But at the moment, we're just looking at relative viscosities between the same protein in different formulations. That's the application that we've developed, and that is, guaranteed to work essentially.
Wonderful. And do you have to normalize for protein concentration and buffer pH when monitoring fluorescence?
No. So generally, because we're looking for changes with temperature, for example, it doesn't really matter if the concentrations are different. And especially if you're looking for a wavelength shift, because that's largely independent of concentration anyway. So unless the conformation changes with concentration, then yeah. But in general, no. There's no need to normalize for protein concentration.
Thank you. And does this work on opalescent proteins?
I imagine so. So we've tested it on things where we've got large, heavy particles or cloudy solutions, and they've worked okay. We've done things with aluminium suspensions or liposomal adjuvants and various things which are fairly opalescent. So very probably yes, I believe it does.
Wonderful. And what is the dynamic range of the unit?
In terms of light scattering, well, we can see changes from, as I say, 0.05 mg per mil. We can see if you go from 100% monomer to 100% dimer, you can see a doubling of the signal there. And we can measure all the way up to 150 mg per mil in terms of concentration. So the dynamic range in terms of light scattering is huge. And in terms of fluorescence, it's probably even larger because it's a very sensitive technique, and we can modulate the signal to make sure it doesn't go too high, for example. But in terms of an absolute number, I don't have that right now, but that's certainly something that we can communicate over the next day or two.
Excellent. And we have a longer question here. In your first case study, you've correlated the melting temperature of the Fab region of this mAb with aggregation over a period of time, and also in the second case study where you have stored at low pH. Have you done any measurements of activity to see if that changes also?
Good question. So the binding activity for this IgG for its antigen would be the best way to determine the effects of storage on function. Now, as I said in the slides, we were given this mAb, which is very kind, and we don't know its cognate antigen, so we've not actually been able to do this additional assay at this stage. But yeah, that would be the thing that you would do, is see how the activity was affected as well, and that would be part of a standard test when you're doing accelerated stability measurements.
Excellent. Well, thank you, Dan, for all of those insightful answers. Unfortunately, we are out of time. So I want to thank the audience for their interesting questions, our speaker, Dan, for his informative presentation, and our sponsor, Unchained Labs, for making today's educational webcast a valuable experience. This webcast will be available for on-demand viewing through December of 2016. You will receive an email from BioPharm International alerting you to when the webcast will be available for replay. So please invite your colleagues who may have missed this live event by forwarding them the announcement. And thank you again for attending today's webcast. We'll see you next time. Goodbye.